Hello and welcome back. Thank you for watching my videos and supporting my channel. So let's begin with this video in which we will learn more about the bacterial cell. All bacterial cells are prokaryotic cells defined traditionally by the absence of a nucleus in it. This definition has been challenged by some microbiologists who are more interested in describing what it has and not merely by the absence of a nucleus. So, in this video, we will study the structure and functions of a bacterial cell from the eyes of a microbiologist in detail. A prokaryote is defined firstly by the way its DNA is packaged. DNA is lying free in the cytoplasm and not inside a nucleus. Second is the structure of its cell wall. Prokaryotes or bacteria have a distinct cell wall that is sturdy and made up of proteoglycans. Finally, its internal structures are also lying free in the cytoplasm and it does not have any membrane-bound organelles like the eukaryotes. Human infections can be caused by both prokaryotes and eukaryotes. An example of prokaryote infection is a boil in the skin caused by Staphylococcus aureus, and for eukaryotic infection is the malaria caused by Plasmodium. The drugs effective against prokaryotes are not effective for treatment of eukaryotic infection. That is why it is important to differentiate between the two. Now let's study the prokaryote form and function in detail. It is generally believed that prokaryotes were the very first cells to be born on the earth. The fact that they have survived for so long in a variety of habitats tells a lot about their cell structure and function that makes them amazingly versatile and adaptable. General cell organization of a prokaryotic cell is as follows. External components of a prokaryotic cell can be either appendages or Glycocalyx. The appendages include flagella, Pili, Fimbriae, and Glycocalyx includes the capsule and slime layer. The cell envelope components include outer membrane, cell wall, and a cell membrane. The internal structures include cytoplasm, ribosomes, inclusions, nucleoid or chromosome, plasmid actin cytoskeleton, and endospore. Now let's study these structures and their functions one by one. Beginning with the Glycocalyx, which is a coating or a layer of molecules external to the cell wall, visible in this picture as hair-like bristles surrounding the cell membrane. It serves protective, adhesive, and receptor functions. It may fit tightly, for example, capsule, or may be very loose or diffuse, for example, slime. In addition to Glycocalyx, other external components of a bacterial cell are the appendages, that includes Flagellum, Fimbriae, and Pili. Flagellum are the specialized appendage attached to the cell by a basal body that holds a long rotating filament. The movement pushes the cell forward and provides motility. Fimbriae are fine hair-like bristles extending from the cell surface that help in adhesion to other cells and surfaces. Pili is an elongate, hollow appendage used in the transfer of DNA to other cells. Now coming to the components of a cell envelope. It includes cytoplasmic membrane, which is a thin sheet of lipid and protein that surrounds the cytoplasm and controls the flow of material into and out of the cell pool. Peptidoglycan-rich cell wall is a semi-rigid casing that provides a support and shape to the cell. Outer membrane is an extra membrane similar to the cell membrane but also contains lipopolysaccharide. It controls the flow of materials and portions of it are toxic to mammals when released. It's seen commonly in the gram-negative bacteria. Now coming to the internal components. Cytoplasm, ribosomes, bacterial chromosome or nucleoid, inclusion bodies, and plasmid. Cytoplasm is a water-based solution filling the entire cell. Ribosomes are the tiny particles composed of protein and RNA that are the sites for protein synthesis. Bacterial chromosome is composed of the condensed DNA molecule, and DNA directs all genetics and heredity of the cell and codes for all proteins. Inclusion bodies or the granules are the stored nutrients such as fat, phosphate or glycogen deposited in the dense crystal or particles that can be tapped into when needed. Plasmids are the double-stranded DNA circle containing extra genes. In addition to these, some bacteria show endospore, which is a dormant body formed within some bacteria that allows for their survival in adverse conditions. Another internal component is actin cytoskeleton, which are the long fibers of protein that encircle the cell just inside the cell membrane and contribute to the shape of the cell. Now let's cover the bacterial components in detail. First, beginning with the appendages. Appendages are divided into two main types, those that provide motility, that is flagella, and those that provide attachment points or channels for material transfer, for example, Fimbriae and Pili. Flagella are also called as bacterial propellers. It has an amazing construction unique in biological world. Its special architecture has three main parts, the filament, the hook or sheath, and the basal body. Filament is a helical structure made up of proteins. It is attached to a curved hook. The hook is attached to the cell by the basal body that is made up of a stack of rings firmly anchored through cell wall, cell membrane, and outer membrane. This arrangement allows flagella to rotate 360 degrees and its motion propels the bacterium towards a particular direction. All spirella, half of bacilli and few cocci are flagellated. The flagella can vary in number and arrangement around the cells. They can either have polar arrangement at one or both ends of the cell or peritrichous arrangement. That means the flagella randomly arranged all over the surface of the cell. Polar arrangement can be further described as monotrichous, that means single flagellum at one pole. Lophotrichous means a bunch of flagella from the same site. Amphitrichous means flagella at both poles of the cell. Flagella are difficult to visualize with light microscope, but presence or demonstration of motility is important for diagnosis of bacteria. Various methods for demonstration of motility include a stab of culture into a semi-solid agar. The motile bacteria move away from the stab line and shows a diffuse growth in the medium, while non-motile bacteria grow only in the stab line. Other methods include hanging draw preparation and wet film using a fresh bacterial culture. I have described both hanging draw preparation as well as wet film method in my earlier videos. Links are given above. Flagella can detect and move in response to chemical signals, and this is called chemotaxis. Counterclockwise movement of the flagellum propels the bacterium towards the stimulus, and this action is called a run. Runs are interrupted by tumbles during which the flagellum reverses the direction and causes cell to stop and change its course. The attractant molecule inhibit tumbles and permits run towards the stimulus. Some photosynthetic bacteria exhibit phototaxis, that means movement in response to light. Now, what are periplasmic flagella? These are seen in spirochetes. As we know, spirochetes show an unusual wriggly mode of locomotion. This is due to the periplasmic flagellae or axial filaments, which are two or more long coiled threads enclosed in the space between the cell wall and the cell membrane. These filaments contract to give the spirochetes a twisting or flexing motion as shown in this picture. Now coming to other appendages that are used by bacteria for attachment or mating but not for motility. These are the Fimbriae that are made up of proteins and they look like bristles. They stick to each other and to the surfaces around bacteria. They help in the formation of biofilms and thick aggregates of bacteria on liquid surfaces, rocks and glass. Fimbriae help gonococci to adhere to genitourinary tract epithelium and Escherichia coli to adhere to the intestinal epithelium. Mutant forms of these bacteria that lack Fimbriae cannot cause infection. Another appendage is pilus, also called sex pilus, which is made up of protein pyelin and is a rigid tubular structure. True pili are found only in gram-negative bacteria, where they help in the mating process between cells called conjugation. The pilus from the donor cell combines with the recipient cell, thus providing a connection for transfer of partial DNA from the donor to the recipient. Conjugation using pilus is seen only in compatible gram-negative cells, and conjugation in gram-positive cells involves aggregation proteins rather than the pilus. Now let us know more about Glycocalyx or the bacterial surface coating. Bacterial cell surface is exposed to severe environmental conditions, and Glycocalyx provides some protection against these. It is made up of polysaccharides, proteins, or both. It also helps the bacterium to adhere to its environment. Glycocalyces differ in its organization and chemical composition. Some bacteria are covered by a loose shield, which is called slime layer, that protects them from the loss of water and nutrients. It is called a capsule when it is more tightly bound to the cell and is denser and thicker. Capsules provide sticky mucoid character to the colonies on agar. Let us see some specialized function of glycocalyces. Capsulated bacterial cells have higher pathogenicity because of the protection provided by the capsule against the white blood cells or phagocytes. By escaping phagocytosis, bacteria multiply freely and infect body tissues. The thick white plaque that forms on the teeth comes from the surface slime produced by certain streptococci in the oral cavity. The slime protects and provides niche to the oral bacteria that in time will produce dental caries. Glycocalyx of some bacteria is highly adherent and cause persistent adherence to the plastic catheters, pacemakers, and intrauterine devices. We have covered the external components of a prokaryotic cell. Now coming to the cell envelope, which is the boundary layer of the bacteria. Cell envelope includes the structures covering outside the cytoplasm. Although each layer has a different function, together they act as a single protective unit. The gram-positive cells have two layers, a thick cell wall made up of peptidoglycans and a cell membrane or a plasma membrane. Gram-negative cells on the other hand have an outer membrane, a thin peptidoglycan cell wall and a cell membrane on the inside. The structure of the cell wall helps in determining the shape of the cell. More importantly, it prevents the cell membrane from bursting or collapsing because of the changes in osmotic pressure. The main component of the cell wall is peptidoglycan, that is made up of long sugar chains cross-linked by short peptide fragments that provide strength and flexibility to the cell wall. Most bacteria live in aqueous environment, frequently causing osmotic swelling, and it is the cell wall of the bacteria that keeps the bacterium from bursting. The drug industry has exploited this feature by producing drugs like penicillins and cephalosporins that target the peptide crosslinks of the peptidoglycan, thus weakening their cell walls. Lysozyme, an enzyme contained in the tears and saliva, provides a natural defense against certain bacteria by hydrolyzing the bonds in glycan chains and causing the wall to break down. Compared to the gram-positive cell wall that has 20 to 80 nanometer thick peptidoglycan layer, the gram-negative cell wall has 1 to 3 nanometer sheet of peptidoglycan, giving them more flexibility but higher sensitivity to lysis. Non-typical cell walls include the mycobacteria and nocardia that stain gram-positive because of the peptidoglycan in their cell walls, but bulk of their cell walls is composed of unique lipids called mycolic acid. This thick, waxy layer of mycolic acid makes them resistant to chemicals and staining with dyes. We use hot carbylfixin dye to stain these bacteria, and the dye binds tightly to the mycolic acid, resisting decolorization with acids. This is the basis behind acid-fast staining used to diagnose mycobacteria and nocardia. Now let us see some cell wall deficient bacteria. Mycoplasma are the bacteria that naturally lack cell wall, but their cell membrane is stabilized by sterols and is resistant to lysis. They are tiny pleomorphic cells ranging in shape from filamentous, coccus or donut-shaped. Mycoplasma pneumoniae adheres to the respiratory membrane in humans and cause atypical type of pneumonia. Some bacteria that normally have cell walls can lose them either by mutation or exposure to lysozyme or penicillin. These cell wall deficient forms are called L forms. L form of gram-positive cell is called protoplast, and L form of gram-negative cells retains its outer membrane and is called a spheroplast. Gram-negative outer membrane is similar to the cell membrane, except that it contains special lipopolysaccharide, also called LPS, and proteins. The polysaccharide chains extending from the surface acts as receptors and antigens, and the lipid portion of lipopolysaccharide is called endotoxin because it stimulates fever and shock in gram-negative infections like meningitis and typhoid. Now coming to cell membrane. Cell membrane is present just below the cell wall. It is a phospholipid bilayer membrane which is a flexible sheet covering the cytoplasm. Phospholipids make 30 to 40% of the membrane mass and proteins contribute 60 to 70%. Major exception to this formation is the cell membrane of mycoplasma that contain sterols, the rigid lipids that provide stability to the cell membrane. Because prokaryotes have none of the eukaryotic organelles, the cell membrane provides a site for functions such as energy reactions, nutrient processing, and synthesis. It also regulates transport and is a selectively permeable structure. Another function is secretion of metabolic products into the extracellular environment. Most enzymes of respiration and ATP synthesis reside in the cell membrane since prokaryotes lack mitochondria. Enzymes in the cell membrane synthesize macromolecules to be incorporated into cell membrane and appendages. Till now we have covered the external components and the cell envelope. Now coming to the contents of cell cytoplasm. Inside the cell membrane there is a gelatinous solution referred to as cytoplasm. It is another prominent site for many of the cell's biochemical and synthetic activities. It is made up of water, which is 70 to 80%, that serves as a solvent for sugar, amino acids, and salts. These components are the building blocks for energy and synthetic activities. Cytoplasm also contains discrete bodies like chromatin, ribosomes, granules, plasmids, and actin or tubular strands that form cytoskeleton of the bacteria. Bacterial chromosomes are the sources of genetic information. Most bacteria have a single circular strand of DNA called bacterial chromosome. In bacteria, the DNA is not enclosed inside the membrane, so it does not have a nucleus. Instead, the DNA is aggregated in a dense area called nucleoid. DNA is tightly coiled around some basic proteins so that it can fit inside the cell compartment. It contains the genes for bacterial maintenance and growth. Some bacteria contain other pieces of the DNA called plasmids that are not essential for their survival. They exist as tiny circular structures of DNA. They can get integrated with the DNA of bacteria and also get transferred to other bacteria through conjugation. They contain special information that help bacteria to have properties like drug resistance, toxin production, and enzyme production. The plasmids are easy to manipulate and therefore are used in genetic engineering for various purposes. The ribosomes are the sites of protein synthesis. Prokaryotes have thousands of tiny ribosomes made of RNA and proteins. When in chains, they are called polysomes. Prokaryotic ribosome has been given a rating of 70S based on its molecular size and various parts. In comparison, the eukaryotic ribosomes are 80S. This is important in identification of bacteria. Now coming to inclusions or granules that are storage bodies for bacteria. Availability of food for bacteria is variable. So during nutrient abundance, some bacteria can lay down nutrients intracellularly in inclusion bodies of various size, number, and content. These nutrients are then used during scarcity. The contents of inclusion bodies include energy-dense glycogen and beta-hydroxybutyrate. In aquatic bacteria, a unique inclusion is found that contains gas and helps the bacteria float. Other inclusions, also called granules, are crystals of inorganic compounds, for example, sulfur granules of photosynthetic bacteria and polyphosphate granules of Corynebacterium and Mycobacterium. The cytoskeleton is formed by long polymers of actin and tubulin proteins arranged in helical ribbons around the cell just under the membrane. These are found in rod-shaped and spiral bacteria. They contribute to the cell shape, influence the way peptidoglycan is arranged, and also help in cell division. Now coming to bacterial endospores that are extremely resistant stage of bacterial life cycle. Endospores or spores can withstand hostile conditions and facilitate survival. They are formed by Bacillus, Clostridium, and Sporosarcina in the presence of adverse environmental conditions to continue the vegetative state of these bacteria. Features of spores, that is, size, shape, and position in the vegetative cell helps in identification of bacteria. Most medically important sporulating bacteria are gram-positive. Endospore formation occurs in the presence of inadequate carbon or nitrogen source in the environment. The vegetative cell first gets converted into a sporangium. Complete transformation into sporangium and then into endospore requires 6 to 8 hours in most spore-forming species. Bacterial endospores can withstand extremes of heat, drying, freezing, radiation, and chemicals. High content of calcium in the spores remove the water and makes them resistant to heat because destruction by heat requires certain amount of water in the cells. It is metabolically inactive and highly resistant to drying. The thick impervious cortex and spore coats prevent damage from chemicals and radiation. More recently, microbiologists unearthed a viable endospore from the 250 million year old salt crystal. Germination of endospores. Breaking of dormancy and germination happens in the presence of water and specific chemical and environmental stimulus. Once initiated, it completes rapidly in 1 to 1.5 hours. Common germination agents include amino acids or salts which stimulate formation of hydrolytic enzymes that digest the cortex and expose the core to the water. As the core absorbs water, it grows out of the spore coat and reverts to fully active vegetative cell. Now, what is the medical significance of spores? Bacillus anthracis is an ideal agent of bioterrorism in its spore form. Clostridium tetani spores cause tetanus. Clostridium perfringens spores cause gas gangrene. Clostridium botulinum causes botulism. Spores inhabit soil and dust. Therefore, it becomes essential to ensure destruction of spores where sterility is required, especially for surgical procedures and implantable devices. So that was all about the bacterial cell. Hope you liked the video. Please like, subscribe, comment and share. Thank you for watching.